you get a half sheet, number 1 to 8, I'm going to start off with seeing if you can identify meiosis. Now remember, meiosis has 2 sets, 2 rounds, meiosis 1, meiosis 2. What we had talked about is prophase, metaphase, anaphase, telophase. In both of those, if you see one cell becoming 2, you're somewhere in meiosis 1, for which phase? If you see 2 cells becoming 4, you're somewhere in meiosis 2. So I randomly mix them up. On your half sheet, I'll be looking to see if you can identify these 8. So title it meiosis, and number 1 to 8, and write the answers on your half sheet. I will not reveal the answers. I think you should know this. I'll give you a couple minutes. I'll give you 1 more minute. Yeah, just telophase is fine. That's what it is. All right, moving on. But one thing I want to say is, if you're having trouble with that, we have a set here in class that I use for the lab practical, which is similar to the pictures that you studied. And this one I keyed out. Okay, so let me teach from this one. And they skip some of the phases in anaphase 2. Okay, but they have many phases that are in prophase 1, early to late. So let's say I pop 2 of these off the board and I randomly mix them up. how can you tell what's earlier and what's later in prophase? One clue I look at is, where are these yellow things? Sensory zones. See how they get further apart as you go to the polar ends? That's one clue I look for. Okay, that's probably the easiest clue. And if you see something like this, oh, you remember, oh yeah, it's anaphase. How do you remember if it's 1 or 2? Well, if it's one cell, it looks like it's becoming 2. It's anaphase 1. But this looks like it's already 2 cells and it's becoming 4. It's anaphase 2. And the other clue is, I always ask, for anaphase, what's being separated in anaphase 1? That's a double. That's a double. These were the homologous chromosomes being separated. Okay. And why is it like this chromosome has some blue mixed up with the red, and this one has some red mixed up with the blue? Remember what happens over here? It's kind of genetic recombinations. That's shown there too. So all of it that I went over in lecture, step by step, it is illustrated on this model set. Okay. Like for example, that's a single chromosome. That's a single chromosome. You're separating the sister chromatids. Another way I'll check you on meiosis today is we're having a lab. And I'll have you draw out and illustrate each phase. Yeah, question?
So that's sister chromatids at the very end?
You're separating the sister chromatids here.
Okay.
Okay. And after you separate them, just call them chromosomes.
Oh, okay.
Yeah. All right. Well, next up is meiosis with the ovarian cycle. So I'm going to teach the ovarian cycle twice. Here's a picture of it. The first time I'll teach it, I'll teach alongside with it in parallel, literally, what's going on in meiosis in the egg. Okay? All right, so let's do that. So if you're going to ovulate an egg on day 14, let's go all the way back to before a female was born. The name of the cells that are the eggs are the oogonium before birth. Oogonium. That's singular. Oogonium is plural. And basically, these are the eggs. And before birth, they undergo mitosis. This is regular cell division. The way I had said in the previous lecture is that the mitosis is you're trying to multiply as many eggs as possible. But that by the 7th month of pregnancy, you get the follicle formation. And I've taught this before. It should be ringing some kind of a bell in your head. The follicle formation that forms are the primordial follicles. Okay? So maybe by birth, you have something like 1 to 2 million primordial follicles. All right, so let's start to talk about the oocyte that's inside that primordial follicle. That was just kind of a recap. If it's not a recap and it's for the first time for you, fine, but this is where we're picking it up. A female is born, and there's 1 million follicles. And today, I have to teach reproduction, so it's like baby girl has to become sexually mature. And at birth, in terms of meiosis, in terms of follicles, 1 to 2 million of the primordials. Did I describe the primordials as kind of a mature or immature follicle? Do you remember that? Immature, and then they don't secrete any kind of a sex hormone, right? We identified them under the microscope. Well, what I didn't mention before is, so we know that the follicle is a cell, and inside is an oocyte called a primary oocyte. So you know, it's surrounded by a thin layer of cells. But inside that cell is a nucleus with the chromosomes in it. The name of the egg is the primary oocyte. And in terms of meiosis, it's not going to proceed past prophase 1. So I'll write that over here in this column. And in prophase 1, the DNA has already replicated and went through all that. It's diploid, it's replicated, it's 2NR. Okay, and I kept emphasizing the number 23. Okay, and I said there's 23 tetrads in this phase. So you can see why I taught meiosis first. What the heck is 2NR, 23 tetrad? That doesn't mean anything to me. And if you didn't study, you're probably saying that right now. Well, you'll have some catching up to do. That's where we are, okay? So let's skip from birth to puberty. Well, by puberty, average age of a female, something like 12.5 years, you'll start to go through a regular ovarian cycle, okay? So maybe have your first ovarian cycle. And so what we're going to do is we're going to select maybe a cohort of 20 follicles from your bank of follicles. Now we skipped 12 years, right, from birth to puberty. There's some attrition here. What I had said previously was maybe that number goes down to 400,000 primordial follicles. Now, on average, you only ovulate one of those per month, but in a cycle, you'll select a cohort of 20 from that bank of 400,000 that will undergo further maturation, further follicle development. Select a group, a cohort of something like 20 primordial follicles. Okay, so you're going to cycle them through, and again, the cycle is how many days? 28 days. About 28 days. Could be 21 days, could be 35 days, but on average, something like that. And that's what we'll teach based off of that kind of monthly cycling. Primordial follicles will proceed through all the stages of development that we talked about before. Here's a picture of a primary. Remember that? You have the simple cuboidal cells. They surround the oocyte. There's the nucleus. You can see the chromosomes inside there. Now we start to get development because the hormone FSH is developing the follicles. Okay? So in a 28-day cycle, this first part, when you're first starting to develop your follicles, it's actually called the follicular phase, okay, of a 28-day cycle. So I'm going to note that. And you start to get past primordial, and maybe you can see some primaries. And I had some of you identify it in the microscope. Well, anyways, once you start to get development, in terms of the primary oocyte, so it's called a primary follicle, but the name of the egg inside, it's the, I'll say it contains primary oocyte. And why the term primary? How many rounds of meiosis? 2. Primary, secondary. There's a for meiosis 1, meiosis 2. So these primary oocytes in the follicle are still in prophase 1. Okay, the whole thing, I'll just keep writing it. 2NR, and they are basically have 23 tetrads. So that's the phase of meiosis in that primary oocyte. This is still a sexually, it doesn't secrete any sex hormones, that's what I'm trying to say. Now when you progress to the secondary follicle, when you start to see other things that indicate it's going to start secreting hormones. So remember, when you go talking about a cycle, always keep in your head. We selected how many to develop? 20. Okay, so maybe 20 will reach this phase. And let's write some of the things I noted there. I talked about FECA before. Something that's not noted, but I want you to add it, are the Granulosa cells. What I had said is that, again, under the influence of FSH, you're going to start to secrete some hormone. Okay, so go back and review your notes on that. Well, one thing I didn't mention before is the Zona Pellucida. It's kind of like an eggshell around the primary oocyte. Okay. It's a glycoprotein shell around the primary oocyte, the egg. Well, it contains proteins like ZP1 through ZP4. Some of these act as sperm receptors, so they have some important function there. So that's why I'm mentioning things. So these are the types of things you see in the more mature follicles. We're starting to get estrogens. So basically these follicles, they start to secrete estrogens. This is the endocrine function. Okay.
Going around the primary oocyte, does it persist into the secondary follicle?
Yeah. So the primary oocyte, you still call it primary oocyte, and you still call it a secondary follicle. You don't call it a secondary oocyte until you finish meiosis 1. And that's almost about to happen. So over here, next to secondary follicle, what's going on is you're still in prophase 1 for all of the oocytes that are in these follicles. So again, prophase 1, diploid, replicated, 23 tetrads, that whole deal. Now here's even a late secondary. Okay. Let's go to the top of the board here. We're still in the follicle phase. Iterative verb. So I'll just write late secondary follicle to match my slide. I mean, you still have the same things. Theca, Granulosa Cells, Zona Pellucida. Well, the thing I want to write here that's emphasized, you have these 20, but one achieves dominance, basically, and selected to undergo further maturation to Graphian. One, there can only be one. One follicle achieves dominance. Well, it can be more than one. You can have a couple ovulations. How often do you have twins? How often do we see that? It's pretty rare. So basically, I normally teach there's one that achieves dominance that will ovulate. So of the 20, one achieves dominance. It's probably the one that has the most FSH receptors. Okay, and you know, I'll note that has the most FSH receptors. I mean, the ones that have the most sensitivity to the gonadotropin, and they're going to be able to proliferate the granulosa cells more, and they'll get to this late secondary phase. You'll get more estrogen. Well, anyways, the reason why I mention this is the one that achieves dominance will complete meiosis 1. This follicle will complete meiosis 1. So we were stuck in prophase 1 the whole time. So you're going to go through all the steps. Prophase 1, then metaphase 1, then anaphase 1. What do you separate in anaphase 1? Homologous chromosomes, the dyads. You just split them apart. And so one cell becomes 2. However, in the female, it's not a very equal cytokinesis. You're going to put all the cytoplasm with the ovulated oocyte, and you give the other cell nothing. Okay, so I'm going to draw like an uneven pinching off. Put all the stuff with the aim that's going to separate. So, you know, let's remember, I kept drawing the nuke, and remember how I drew the chromosomes? I used purple and green, didn't I?
Yeah.
So I think I went like long purple, short purple, but it's doubled, right? These are dyads that you've separated. So you have one here, and then in the cell that gets nothing. Long green, short green. You're just pinching it off. Okay, and that's kind of the completion of meiosis 1. You just kind of, let's pretend we finished the pinch off there. Okay, and that's going to split off. And okay, once you complete meiosis 1, now you call this cell not primary oocyte. Now you start calling it a secondary oocyte. Secondary oocyte. And you call this cell the first polar body, because there will be a second. So one cell became 2. Secondary oocyte, because now we're going to be in meiosis 2, and you know, first polar body. So that's kind of, that starts to happen when this one follicle that will become Graphian, that's when you complete the meiosis 1. All the other ones, the other 19 secondaries, they're just going to like keep secreting hormones, and they'll never finish the meiosis one thing. So any questions on that? I'm gonna proceed. So identify follicle, Graphian. They call it mature vesicular. That's the same thing. We call it Graphian. You can see the oocyte right there. Now we call it not primary, but secondary. So let's see what I see on the slide here. The one selected follicle is a Graphian or vesicular follicle. Meiosis once completed. Now the oocyte is called secondary oocyte. So now we're in meiosis 2. But, the secondary oocyte begins meiosis 2, but it doesn't finish. It becomes arrested in metaphase 2. Okay. All right, hold on here. So I'll kind of leave that. So this cell, secondary polar body, it's split off. I only drew 2. But when you're a secondary oocyte, I drew 2 dyads, but really I'm reminding you it was 23 dyads. Okay, maybe I should write that. And you remember how we called it? That's not diploid, it's haploid. But it's replicated. A dyad is a double. It's replicated. That's why we need this second round of meiosis. Okay, so now we got 2 cells. An angry little polar body over here because it got nothing. This got all the cytoplasm, but it got the same amount of genetic material. So this is kind of where we are. We're in a Graphian follicle here. So where we're going is we've got to have ovulation. So this Graphian follicle has to ovulate, which hasn't happened yet. So now we're in meiosis 2. We have a secondary oocyte. You'll start it, but you won't finish it. The secondary oocyte will become arrested in metaphase 2. All right, that secondary oocyte does not finish. Meiosis 2. It becomes arrested in metaphase 2 or anaphase 2, depending on what book you're reading. It doesn't matter. The point is, it's not finishing. Because you don't want to finish. Remember, the whole goal is fertilization, reproduction. And if sperm isn't present, it's not gonna. And so there's some kind of cell-to-cell communication between maybe granulosa cells and other cells within the follicle. I'm not sure what the crosstalk is. I just want us to know you don't finish unless sperm is present. So both of these two cells, and that's not even called a cell, they call it a polar body. They're starting to finish and they just kind of get stuck. So,
and the polar body still doesn't have a cytoplasm, right?
Yeah, it's got nothing. You're right. So let me kind of draw that. Here's the polar body. And we kept drawing that spindle apparatus thing. That whole thing, whole spindle apparatus. And then for the secondary oocyte. stuck in metaphase or anaphase 2. So I'm drawing metaphase as an example. Okay, still with the 23 diaz things, but the point is that you're just kind of stuck there. You're waiting for sperm to arrive. So this is the follicle that will ovulate. And this is where we're stuck. Okay? Okay, so ovulation, D-Day, day 14 of the 28-day cycle. So ovulation marks the end of the follicle phase. Ovulation of what? It's a secondary oocyte into ampulla of uterine tube. So I'll put in parentheses, marks end of follicle phase of our 28-day cycles. It's day 14. I need to remind you of that. Day 14. Marks end of follicle phase. Okay, so that's what you ovulate. I'm not even sure what happens to the polar body. Just let's focus on the ovulated oocyte. So after ovulation, there's a picture of it there. That's a nice real picture. And one of our colleagues circulated this real picture. Person was in surgery and happened to catch the ovulation happen right before their eyes. Someone got out a camera and took a picture of it. And you can see the follicle, the egg coming out, and it's for real. It's a very large cell. It gets ovulated and swept up into the oviduct. And that brings us to the second half of the 28-day cycle called the luteal phase. So I'm going to get rid of follicle phase up here. So the luteal phase is the other half, you know, say 15 to 28, those days. Ovulation was day 14. Basically, you have first half follicle phase, ovulation, then second half luteal phase. So when you ovulated, the egg was ovulated into the uterine tube, but what was left behind in the ovary is a ruptured follicle. The ruptured Graphian follicle bleeds. And there's a name for that. It's called corpus hemorrhagicum. I've gotten tired of spelling that. So that means bleeding body. So let's just call it that. The ruptured Graphian follicle that just now ovulated is called a bleeding body. Bleeding body. And what happens is there's the hormone of the luteinizing hormone, FSH-LH, the LH part, is going to target that bleeding body and turn it into an endocrine structure called a yellow body. But I want you to call it the corpus luteum, which means yellow body. And because that's why we call it the luteal phase. I mean, this hormone, the hormone this secretes is called progesterone. Okay, so more on that later. But what I want to focus on here is the pictures anyways. That's a picture under the microscope of corpus luteum. That's from the book. This is a picture I took from our collection. It's not much to look at. It's just, yeah, I don't think it's much to look at, but it's more interesting for its function. It secretes progesterone. I don't remember that. Okay. Well, I got a couple of hormones here, don't I? Remember that? Is LH tropic or not? It's tropic. It's causing the release of this. Okay. Progesterone is a hormone too. So let's get back to meiosis. So this is in the ovary, right? But this is not. This is in the ampulla of the uterine tube awaiting fertilization. Okay. So let's say you're not pregnant. Well, nothing happens. This just never finishes. Let's say sperm is present. I got a little break here. Let's just go. Sperm body. Sperm got in, fertilizing sperm. What did the sperm contribute? Just the genetic material, haploid. Okay. So I'm going to just draw, I don't know, about two blue ones, one long, one short. That's the sperm contribution right there. So now you can complete meiosis 2 if sperm is present. Now I say if sperm is present, but where are we anatomically? Typical site of fertilization. It's on somebody's quiz. Ampulla of uterine tube. Okay, so that's where we are in the body. All right. So how can I draw this? So to finish meiosis 2, this polar body, it may divide into two. Okay. Let's see here. So that may split. So that one cell became two. I'm going to draw one long and one short green in each, right? You finished meiosis 2. So these two polar bodies came from the first polar body. I'm going to still label this cell first PB, polar body. But it underwent its finished meiosis 2 thing. Okay, but we don't care about the polar bodies. This is what we care about. Okay. So maybe we can go like this. So we're going to split the doubles there. Half will go to the sperm. The other half will inject it into a second polar body. So I'm going to go like this. I'm going to draw like that. So sperm got in. The winner, all the millions of sperm. A typical ejaculate, I said, has maybe 100 million. 100 million, but that's in the fornix. By the time you swim to the oviduct, maybe you're down to thousands. Okay, and the first ones that get there, they're not the winners. They're just punching holes in the outer layers, like through Zona Pellucida. And then maybe a second wave, the winner. Okay, and so his contribution again was 23 single chromosomes. I'll just draw two, one long, one short. And what happens is you split that apart. I'll draw one long purple, one short purple. I mean, they kind of go towards the male single chromosomes, and the other ones one long, one short. They're going to be pinched off into a second polar body. So how many polar bodies we got? 3. Those 2 ones came from the first, called this second polar body. Don't care about it. Okay, so this is the thing we care about. That's the fertilized egg. We'll get more into that later. So this is, we call it conception, when the chromosomes from mom and dad intermix. But more on that later. That kind of finishes meiosis, right? When you have sperm present, you finish meiosis too. Well, what happens after the luteal phase, if you are pregnant, let's do that scenario A. You are pregnant. It's binary. You are or you aren't. So option A, are pregnant. This is repro. That's option A. Yeah, look, that's what happens. What happens here? This corpus luteum remains and will continue to secrete progesterone. If pregnant, I'll just say remains. Continue secreting progesterone. Well, progesterone, it targets the endometrium. As long as you got progesterone, you're going to miss menses, okay, to support the pregnancy. So I want to write that.
Is that what's tested in the pregnancy test?
No. Actually, what they test for is when this implants in about a week, it will secrete a hormone called HCG.
Oh,
That is what they test for in the urine test, home pregnancy test. But you'll miss menses, and everyone knows, oh, you missed your period, maybe you're pregnant. And everyone knows that, but this is why. This is hormonally why. Because in pregnancy, that structure, the corpus luteum, remains. Normally it has a 2-week lifespan, but if you're pregnant, it'll hang around for a few months until the placenta gets in place. Okay? So that's option A. So option B is not pregnant. That's a slide. Yeah, that there. Option B, not pregnant, no sperm. Well, you're just stuck there, you never finish. Okay. And whether or not this completes the second division, who cares? This is repro, there's no sperm, not pregnant. What happens is corpus luteum dies. It's got about a 2-week lifespan, and then you'll start menses. Well, this structure becomes it shrivels into a white scar called the corpus albicans. Shrivels to a white scar called corpus albicans, which means, white body. A little bit of scar tissue at the end of each cycle that you weren't pregnant. That's what it looks like under the microscope. Yeah, well, that's just scar tissue. It was secreting progesterone, but when it dies, no progesterone. So I'll note for you, stops secreting progesterone. And what I had said is, let me write this, progesterone. When you stop secreting the hormone, there's nothing to maintain the endometrium, so then you have menses. So this will be about the 28th day of the cycle. D28, start menses. So that basically, I taught this figure. So what you should do is like go through your notes, go all the way back to meiosis if you're fuzzy on all the terms with meiosis, and then go through these notes from this morning and start. This is why I started. Oogonium, mitosis, growth, it's before birth, right? And infancy, childhood. I mentioned the primordial follicles. I mentioned this. Now, I didn't use their terminology. I didn't say 2N. I said what? 2N R. I want you to remember, it is replicated. Okay, other books use 2N R, and I kind of looked at that from another book. But it is diploid, but it is also replicated. And then each month, from puberty to menopause, I talked about that, and that's when I started talking about when you progress past primordial, primary, secondary, vesicular. You see when you get to vesicular, how they parallel it with the oocyte, the primary oocyte, it's going to start to spit out the first polar body. One cell becomes two. This cell kept all the cytoplasm. We now call that secondary oocyte, and that's the first polar body. Now, I wouldn't call that N. I would call it NR, because what I've noted for you is that in each of these cells is 23 dyads. It's still replicated. So you do your second round. If sperm is present, okay, you ovulate, obviously, sperm's there, and you get your second one. And you get 3 polar bodies. One's the second polar body, and these 2 are from the first one. And then and it has everything else. And you ovulate. I call that a bleeding body. That's the yellow body. And it degenerates if you're not pregnant. But, so, it took me a while to teach this. So go back and review that. And I have some questions here. I don't want to answer these now. I feel like I'm on a roll. And I want to take you right to the meat of today's lecture. I'm going to teach the ovarian cycle again, but this time I'm not going to emphasize meiosis. I'm going to emphasize the uterine cycle with it, along with an emphasis on these hormones. Okay, and I think that's really important. If you understand this, I think you'll have a full understanding of reproduction. So what I'm presenting now, this will be the written portion of lecture exam 2. So maybe draw a big line in your notes. Essay. Use capital letters. Usually when I have students write about this, I make it worth 25 points. So students who decide not to do it, then you can't get an A. That's why I stack it. And how I kind of go through it is the 28-day cycle, but I try to emphasize in terms of the anatomy and physiology, the hypothalamus, HYP, because that's where, the hypothalamus / ant pit, that's where you're getting your gonadotropins. In the middle, I always like to put the ovary, because the gonadotropins have to target the ovary, but then the ovary, when they secrete estrogen, progesterone, they have to target the uterus. And let's be specific. It's the functional layer of the endometrium, right? There's my shorthand for functional. Functional layer it thickens, it sloughs off, and that's at the bottom. The uterine cycle is at the bottom. And at the top, well, they have, I put gonadotropic. I actually added that in because they didn't have that. So let's start at the end of the previous cycle where you weren't pregnant. Okay, right here. So do you see how they have it color-coded from top to bottom is light blue? That's supposed to be for menses. And so menses, so you weren't pregnant. So what happens is there's very low estrogen and progesterone. Progesterone. Let me just write it out together. There's low estrogen, progesterone. That causes menses because it's the sex hormones that thicken the endometrium. So when those levels crash, what happens is those spiral arteries, they start to spasm. It cuts off the blood flow, and then the tissue is sloughed off. Spiral arteries, spasm, endometrium sloughed off. That's menses. So it starts at day 28 to day 5 of the next cycle. Okay, so that's what's going on here. So on the figure, I mean, I would like you to see everything that's being pointed out there. I said there's low estrogen, progesterone. So on day 28, they do show that these hormone levels are dropping, and that you've thickened, but you're not pregnant, so now you just kind of slough it off. And it proceeds over here. Okay. So let's start over in the next cycle. So what you have to look at is the top of the figure, the GnRH. It slightly starts to increase in pulses. It's secreted in pulses. So what you have to know is that this fact over here, estrogen and progesterone are an inhibition on the hypothalamus. So when you remove that inhibition, low estrogen, progesterone removes inhibition. On GnRH. So you're going to, I'll say, increase pulses. And you can look at the book, it even tells you the number of pulses. I don't think you really have to understand that. Well, what you should know is that gonadotropin releasing hormone releases the gonadotropins FSH and LH. Well, in this part where we're at, now you're going to start to see an increase in the hormone FSH from the hypothalamus and pit. And you can see that on the figure during menses. We're still in the light blue here, first 5 days, and you can see this subtle rise in FSH right there. And that's going to develop your follicles, right? So I'm going to draw a line going from here to here, because you have those, you select a cohort of 20, right, to develop. You go through primordial, that phase, to what? Primary. So these first two, primordial, primary, there's no hormone. But once you get to secondary, those primary oocytes are in there. But anyways, the secondary follicle has those structures that I've been pounded over your head, right? The whole theca, Granulosa cells, and I mentioned ZP, Zona Pellucida, those things you see in the more mature follicles. You know, this is where you should explain the physiology, okay? It's the theca cells when I had said for your previous test, they secrete androstenedione. And then I said that that is a male androgen. It's transferred to the granulosa cells, and under aromatase, it will be converted from androstenedione to estrone. I want to write transferred to the granulosa cells. Okay. I want to write estrone. Okay. I'm going to tag the arrow with aromatase. So this is all under FSH influence. I want to draw arrows from FSH to all of these things. FSH is really causing all of this to occur. The thing about it is, estrone, I want you to understand what it does to the endometrium. Because now we're going to progress past menses and start rebuilding. And we call that the proliferative phase. So I'm going to draw another red arrow from estrone to here under this column, because the proliferative phase, I already said what days that are. So now we're talking from light blue to dark blue. So that's kind of where we are here. We're kind of getting more mature follicles, and we're starting to get some proliferation. Proliferative phase. So I said that was like day 6 to 13. Okay, so that's kind of where we are. But for the first part of that, maybe 6, 7, 8, something like that, you're starting to see, here are the details. You increase spiral arteries. You increase the uterine glands that secrete glycogen. These are the things that proliferate. You're trying to get ready for the luteal phase, for progesterone. So you want to increase the progesterone receptors. You prime for progesterone. Increase progesterone receptors. And the goal of reproduction is to let sperm in. There's normally a mucus plug at the cervix. This kind of dissolves it. Dissolve mucus plug at cervix, let sperm in. Dissolve mucus plug at cervix, comma, let sperm in. Let sperm. So it's the estrogen that causes this thickening here. So after secondary, okay, one thing is, I'm going to use a different color here. Well, this estrone is very important because it caused the proliferative phase, but it provides a negative feedback on the hypothalamus and pit. So I'm going to use this different color here to emphasize negative feedback. You're turning off the original stimulus. You got your 20, right? Turn off FSH. Just note to yourself, we got 20. You don't want any more. That's enough. Just turn you off. So that way you'll stop developing more follicles. You just select your 20 and then stop. So you kind of turn it off. And I want you to see that on the figure. So negative feedbacks are important. There's like 3 of them. This is the first one. These negative feedback. So you get this subtle rise in estrogen, right? Because these vesicles are getting, these follicles are getting bigger. Yeah, the subtle rise. So this first rise here, this first section, there's a drop off in FSH right there. And that's what I'm talking about. Okay, so you're supposed to be able to figure this all out, right? So even me telling you, you still might forget. But that's the first negative feedback. Well, then you go from secondary to graphene, so let's do that. So you have any questions before I erase the board?
From that figure, it looks like the fecal cells and the granulosa are what become the corpus luteum. Is that the case, or is it just a portion of the ovary that injects the
Oh, no, yeah, it's the actual, what you said, becomes the corpus luteum. Yeah, whatever's left of the follicle. All right, so I'm going to erase this. So where we're going with this, the goal is reproduction. So the first thing you got to do is ovulate, but we're not quite there yet. We have our 20 that reached secondary. How many reach graphene? One. One achieves dominance and reaches graphene. Because here they just show one, but really it's like something like 20, that's what I usually read. But one will reach that one. Okay. Okay, one achieves one follicle. Achieves graphene. I'll just write graphene, big letters. So that's the biggest follicle, and you'll see even more of an increase in estrogen levels. Okay, so we're getting closer to day 14 here, but we're not quite there yet. We're still pre-ovulation. But what I'll say here is we'll get even larger increases in estrogens. More estrogen. So you'll continue proliferative phase. I'll just write that. I already gave you all the details I want you to know for that. Okay, the thing about it is a little bit of an increase turned off the gonadotropin, but this larger increase that we see now, that's actually a positive feedback. Use red for positive feedback. I think what I'm doing here, I'm using red for stimulate or positive feedback. I use blue for negative feedback. And so the positive feedback here is you trigger what's called the LH surge. So that's shown by this large spike. It's kind of flat line, and LH surges right there. You also get an increase in FSH, but let's just forget about that. Let's just focus on the LH. That's the important one here. So that positive feedback causes the LH surge. No, exclamation point. Okay, that surge, the LH is going to trigger ovulation. So let me draw an arrow going back from LH to ovary. So this LH surge causes ovulation. Again, day 14, marking the end of the follicle phase of the ovarian cycle. Day 14. If this is the day we were all conceived, right? Maybe the next day, but the point is, now it had to have been that day. Okay? Well, you had to have been conceived, and it had to have been on a day. It's going to be that day. And I'll put fertilization, but I'll put a question mark in case it happened or in case it didn't happen. We'll get back to that. Okay, so let's stick to that. Ovulation is the graphene follicle ruptures. It becomes a bleeding body. I'll say the follicle becomes a bleeding body because the oocyte is gone. not in the ovary, it's in the uterine tube. So not only did LH cause ovulation, it weakened the Graafian follicle wall, the oocyte was allowed to burst out of there. It triggered ovulation, but it also transforms this bleeding body into the corpus luteum, or yellow body. So I'm going to draw an arrow going from LH to here, because it also causes the transformation, I guess I'll put that word, transformation of the bleeding body to the yellow body, which we call the corpus luteum. So this starts the luteal phase. Okay, so we go from follicle phase, ovulation, to luteal phase. And this structure secretes progesterone. It's the major female sex hormone. So what you're going to see here is this progesterone, again, targets the endometrium. And what it'll do is it'll take you into the secretory phase. Causes secretory phase. So now we're post-ovulation. We're into the light green area of the figure, past day 14. And you can see this continued thickening where the endometrium, it becomes a secretory mucosa. Pretty much it's just an environment primed for implantation. One of the things that happens is you create this private uterus by reforming mucus plug at cervix. So that's one detail I'll give you about secretory phase. Reform mucus plug at cervix. Private uterus, because if you fertilized, it happened already. Okay. When did it happen? What day? 14. But we're past that now. Okay. So it's like you're preparing for implantation, maybe 7 to 10 days post-fertilization. Okay, so which would take you right to here. The secretory phase, we'll just look at where the green. I think I called it day 15 to 27, right? That's about what it says here. 15 to 27-ish. Day 15 to 27 post-ovulation. So that's what's going on here. You're ready for implantation if fertilization occurred in the oviduct. But the other thing about progesterone is it's going to cause a powerful negative feedback. Okay, but I'm kind of at the bottom, so I'm going to erase this unless there's questions about this. All right, so let's pick up where we left off. We have increased levels of progesterone. I already told you it causes secretory phase. Or just phase, not phases. And this is really the difference. This word cause is the difference between like an A essay and a B essay. The A student knows what hormones cause what. The B student can remember the steps, but they forget to say which hormone causes this. You'll write the hormone, but then you won't specifically say what it does. It'll just be there, and I'm like, do they know what it does? And I have to guess. And I usually guess by marking it wrong. Okay, so it's very important that you put this causes that. It tells me that you know. Okay. Progesterone also causes a powerful negative feedback on LH. Let me turn this down. It's going to turn off LH. You can see that. The reason why the LH surge is a spike, well, it's surging on one end, that's the stimulus, but the other end, it goes down immediately. And to create a spike, you have a powerful surge, but then a powerful drop. Right? It's a powerful drop bar. It just drops off. And drop off. Okay. So it drops off. So this is the gonadotropin that gave the corpus luteum life. Okay. So now it's kind of like on a lifeboat. It's got a 2-week lifespan. Without LH, corpus luteum has 2-week lifespan. This time, let's do the not pregnant option first. Let's say it does have a 2-week lifespan, because why? There's no LH to sustain it. Okay, so without the gonadotropin to sustain the corpus luteum dies, it becomes corpus albicans. It dies. Well, we call it a white scar, corpus albicans. And I said that structure, it does not secrete progesterone. And what we had said over here is no estrogen, no progesterone, that's menses. Right, so we're right back to day 28. Low estrogen / progesterone, day 28, menses. So we got back to where we ended, so I'm at the end. I just got to talk about that other thing where you are pregnant, okay? And I already mentioned that. But what did I say? Oh, okay, I should mention this. So I'm going to leave that there. So we can move over here. So we're backing up, going back in time, getting back in a time machine, if we are pregnant. Corpus luteum secretes what? Progesterone. Okay, but it's all still true. This still has negative feedback. But now I'm saying we are pregnant. So sperm is present. If fertilization did happen, what day did it happen? Day 14. And if it did implant in the uterus, it might have happened maybe 10 days later. 14 plus 10 is 24. So right around here-ish, so let's say here in the uterus, pregnant, so you got implantation, something like day 24. Okay, something that I'll talk about more in development is the implanting, it's called a blastocyst. Spell that right, blast. Will become embryo, baby, okay? It secretes, it starts to secrete a hormone called HCG. HCG stands for human chorionic gonadotropin. It's coming from a structure that will be the placenta. So it's coming from the pregnancy for sure. And so this hormone, the HCG, okay, that will sustain, it will sustain corpus luteum. Okay? Write that down. HCG sustains corpus luteum. Now, what was the thing that gave corpus luteum life after ovulation? What was the hormone? What was the gonadotropin? LH gave the corpus luteum life. The bleeding body. LH is the starting quarterback in Jimmy Garoppolo. Okay? But then what happened? Progesterone did what? Turned it off. Like Jimmy Garoppolo broke his ACL last year. And then you send in Nick Mullins. That's Nick Mullins, HCG. Okay, it's like, okay, well, now we want to sustain corpus luteum. This gonadotropin will now sustain corpus luteum because LH is gone. Okay, so boom, well, it continues to secrete. I'm sorry if you don't watch, you're not a 49ers fan. The point is, that's not there, now that's there. Not this gonadotropin, but that gonadotropin from the pregnancy. Okay. Continues to secrete. Progesterone. As long as you get that, I mean, you're going to go well past day 28. So I'll go to here. Progesterone sustains endometrium. You miss menses. That's how some of those contraceptives work. They block the action of progesterone, so you have menses whether you have implantation or not. But here in repro, we talk about you sustain it. That's why you miss. And so you get past day 28. So yeah, okay. So we did both option A, option B. Are any questions on what we went through here? I know it was a lot. Okay. Well, what we can do now is take a break. Let's break till about 9:15. When we come back, we'll have a lab on meiosis.